Giovanni, and he is going to present a poster based on a novel CO2 responsive micellar enhanced ultrafiltration for the removal of emerging contaminants from water. So go ahead, Giovanni. Thank you so much.
Thank you so much, Victoria, for the opportunity to share a little bit about my work. So yes, as you've mentioned, this is a project that I've been working for the last couple months. I've been doing this in collaboration with my two supervisors, Dr. Pascal Champagne, which is working at the NRC right now, and also Dr. Philip Jessup. So I like to start talking about my research. Imagine you wake up one day, maybe not feeling so well with a headache, for example. What do? You take a pill or some sort of, medication. But it turns out that most of the medication it actually is not metabolized by your body. It ends up being flushed down, and we thought that those drugs, those chemicals couldn't make no harm to nature, but now we know that they cause a lot of harm. So this is what we call the contaminants of emerging concern. So these are chemical substances, as I mentioned, they were recently discovered. The reason why they are so bad, they bioaccumulate. So after a long time, like small animals somehow intake those drugs, and that in the food chain, for example, it can, like the concentration might rise. And usually these things are not regulated. These chemicals, they are everywhere. They're super bad. So a lot of issues were already reported. I have listed a couple here. Things like increased mortality of fishes, for example. We have a lot of endocrine disruption going on. For example, we have cases of adult males becoming infertile. So the whole idea of this project is to really develop a treatment method for these contaminants of emerging concern. Well, we have water treatment plants that could do the job, right? The thing is, these plants, they were not designed to remove these chemicals specifically, right? So in my research, I am trying to offer a possible strategy that can remove these contaminants from water. And that is what I call the micellar enhanced ultrafiltration. So basically the way it works, we use a special chemical, which is called the surfactant, and we induce the formation of things that we call micelles. These are basically three-dimensional structures that can be formed when micelles organize themselves in some sort of, like sphere kind of thing. So the idea here is that hopefully these micelles, they can favorably interact with these drugs and remove them specifically through some mechanisms like electrostatics. So imagine like a magnet, right? So hopefully our micelles will be able to really absorb the drugs. And then this is kind of like the setup that I've been using currently. So we have a step that we call micellization, which is forming the micelles, and then we let it some time for the removing process to occur, the interactions between the micelles and the drugs, and then we filter. So what I wanted to tell you here, ultrafiltration membranes, they are actually already used in water treatment plants, but they're not used for the removal of small organic contaminants. And that is just because ultrafiltration membranes have large pores. So they are really good to remove large particles, even like microorganisms like bacteria, for example, but they can remove small organic micelles. But when we make those small drugs to bind to something larger, something bigger, that's when we can filter off those things using a ultrafiltration membrane, and this can be done, at really low pressures, which helps in terms of a lot of potential benefits. For example, it's an easy to use setup. Filtration, as I'll show you, it happens really fast. It is cost effective, mainly because of the low pressures that are used, but also because we can recover the material and reuse it again in new cycles, right? And as a whole, I am claiming that my strategy is sustainable because after all, we are removing drugs from water, we are generating clean water for the population through strategy that is sustainable, that can be recovered. Okay, so moving on to some sort of characterization of my materials. So I have some really strong evidences that I have my micelles being formed. So as you can see on the left side there, the left side corner, these are some microscopic images of my micelles. So they were formed. So I've characterized also my membrane. So it was all good. By the way, I am doing all the filtration experiments using a stirred cell. And this stirred cell operates in what we call the dead end mode. So whatever that enters the filter comes out and cannot be recycled into the cell. Well, this is basically because when we are developing such technologies, this is how it works. So if I want to have the thing back again to a new cycle, I need to reopen the cell and set up the whole thing again, but I can explain more about that later. So here I have some results about the flux. So it has a pretty interesting water flux. That is a parameter that is essential for us because population is not going to wait for a long time to get clean water. So we need to really have something that flows nicely because the water, it flows through the membrane and it has to flow at a decent rate. And we had some really interesting results that shows us that, hey, it is working nicely. It has a nice flow. In terms of the drugs specifically, I've been working mostly with antibiotics. So I have a selection of some of those drugs on the left side here. So it turns out that, okay, I'll show you basically, I will call your attention to the blue bars first. So the blue bars, it's the removal by the membrane only. So without the presence of the surfactants. So as you can see, we have some rejection, which is the parameter that we use to assess these kind of strategies, this kind of techniques. As you can see, just the membrane, it does a kind of a not so good job, right? It does reject some even though the pores are big, but some of the drugs might end up being absorbed onto the surface of the membrane. But with the addition of the surfactant, things changes dramatically. As you can see, removal, it kind of rockets in the presence of the surfactant, which is actually our technique. Regrettably, for some of the drugs, it doesn't work so well. Well, we have some technical explanations. So for example, we have the sulfamethoxazole drug here. It's an antibiotic. So at the pH that we were working with, the drug becomes anionic, and our micelles are also anionic. So our hypothesis here to why it didn't work, it's just because we had a repulsion going on. But we are currently working on that to fix that. So this is how the filtrate looks like. I'm doing bench scale experiments so far. So the thing is, it looks clean, right? But it has a lot of surfactant because this is one of the downsides of my technique. Some of the surfactant, it ends up leaking, and no matter what, this is something that it's how it works. But good news are we can recover the surfactant and we can get a new filtrate back again upon the addition of CO2. And that's why CO2 plays a really important role. So basically the way it works, the addition of CO2, we bubble CO2 into the water solution, and it precipitates, actually what it does, it protonates the surfactant. So the surfactant then becomes insoluble in water and it easily precipitates. I have here also some results to show you that, hey, before the addition of CO2, I will call your attention. These are some resonance magnetic results to show you that, hey, the red lines, it's the NMR for this filtrate before the addition of CO2. So it contains all the peaks associated with the surfactant. And after the addition of CO2 and after a simple filtration step, the solution is pretty clean and free of the surfactant. And even if I still have some of the surfactant dissolved, it is not so bad because the thing is a biodegradable material, right? And the LD50 of this material is crazy, crazy high. Just for example, a person to get killed, like an adult weighing 70 kilos, for example, it would need to eat like 3 kilograms of the surfactant. So that is something that I'm pretty optimistic. It's not a big deal. Okay, so as I mentioned, we can reuse the surfactant. So I have some preliminary results here showing you that after 3 cycles of recycling the surfactant, it has a pretty decent performance over these 3 cycles. So I think the material is really good. It does work. And as I mentioned, we had the issue with the not removing the anionic surfactant. I'm sorry, not the anionic drugs. How can we fix that? Well, using one cationic surfactant that could then, attract the anionic drugs. So the idea here is to have like several reactors connected in series, for example. This is something that I would need to discuss with an engineer, for example. But so far, I just have some interesting results that, again, the blue bars, they do represent just the membrane doing the work. As you can see, also now I have here a complex mixture. So I've increased the number of drugs that I'm now working with. So I have 6 drugs, everybody mixed in the same samples, and the surfactant, only the membrane only actually. It doesn't do a good job. Like, as you can see, the blue bars removals are a bit like limited, but upon the addition, upon adding the surfactant, then it becomes like really a big difference in terms of removal efficiency. So that is basically what I have to tell you so far. So we have developed something that is excellent, that is really good for the removal of cationic and neutral drugs. And our technique, it uses mild conditions. So the method, it achieves a high water flux and a fast filtration rate with low pressure, which is something really cool and interesting, especially in terms of the costs associated with this kind of technique. Well, my procedure, it allows the easy recovery of the surfactant. Almost essentially everything is recovered. And this shows, well, yeah, the feasibility and environmental friendliness of the new method. So, and I'm always thankful to my supervisors, as I mentioned before, Dr. Philip Jessup, Dr. Pascal Champagne. I'm also super grateful to the Department of Chemistry for all the help with the facilities and the funding is coming from the Ontario Research Fund. I would also like to give a huge shout to my colleagues in my lab. So yes, that's it. I'm sorry if I rushed about my
You
my
didn't rush
works.
at all. That was excellent. Thank you so much. No, that was a great presentation. It was really,
Thank you.
You did a great job of being able to, well, I could understand it very well.
Oh,
I see
awesome.
that comes with your, vast teaching background as well. I'm sure you're quite good at explaining. So that was really great and so interesting.
Thank
Wow.
you.
How long have you
Thank
been,
you. Thank you.
how long have you been working on this project? Has it been the beginning of your
Yes.
PhD or?
No, I didn't start my PhD with this project. I started doing something else, still doing water treatment research. But when I first started, I was using silica particles. So basically, we in the lab, we have these silica particles. Usually they are used for chromatography or separate purification as a purification technique. So what I was doing, I was functionalizing those particles, functionalizing those their surfaces with CO2 responsive, functional groups, and hopefully again, maybe thinking that this could be helpful and for the sake of removing drugs from water. This project, though, I've been working on it since winter 2023.
Wow.
So it's been
not even a year.
Like a little over a year now. Actually, maybe a year and a half, maybe.
Oh, okay.
Yeah.
Wow. That's a lot of progress.
Yeah, but Victoria, I actually, as a scientist and a researcher, I still, I think I still need a lot to improve here.
Yeah.
Especially, because maybe if that's okay to make an additional comment.
Of course, yeah.
So the reason why I want to work with as much drugs as possible because, in a real environmental sample, you won't find only 2 or 3 drugs, right?
And everything.
Usually it's everybody, like all these bad things together,
Mhm.
mixed, dissolved, whatever. Of course, depending on the region, depending on the country, you might have, a higher concentration of chemical wax, or I don't know. Yeah.
Yeah. No, you're definitely right. It's not just a couple antibiotics in water, unfortunately. But yeah, that's I guess a
Yeah.
lot more testing of things. And yeah, that's great. I mean, I'm sure you were going to get into it, but even, thank you for presenting. I'd love to just even get a little bit more background on what kind of initiated this project. Was it a personal interest? Was it a lab interest? What specifically drew you to this specific project and
Yes.
a bit of your motivation behind it?
Yes, thank you for your question. So, yeah, actually, maybe a little bit more for background.
Yeah.
I'm actually part of a big team at Queens.
Yeah.
So, I'll just come back. So, this ORF, it was a grant that we
Oh, wow.
got back in 2019. I wasn't even here in Canada. And Dr.
Mm.
Pascal Champagne here, she was the PI of this big team. So, basically, I can even send you the links afterwards. It's
Yeah.
called the Contaminants of Emerging Concern
Yeah.
Research Excellence Network.
Yeah.
So we have chemists, biologists, engineers, people from all sorts of backgrounds. And so the big umbrella, basically, we do have three big teams. One does removal techniques, just like myself, right? The second team does detection. So how can we treat the water if we don't detect the contaminants, right?
Yeah.
And the third team, it does work on understanding the fate of these drugs, like the sources. What's the
Mhm.
pathway that the thing goes, like out in nature or something like that? Okay, so
Yeah.
coming back to your question, we were like brainstorming, my supervisors and I, okay, okay. So my first technique, it was working nicely, but I wasn't able to recover the material. And that was something that was really important for this project here,
Yeah.
because we have other things available out there, right? But you like absorbent materials, right? So
Yeah.
you have a solid, the solid can absorb the material quick and easy,
Yeah.
but then the recoveries are usually, they use like high temperatures or the use of harsh chemicals. And as a person who really appreciates sustainability, I wanted to keep away from those things, right?
Yeah.
Okay, so my first materials didn't work so well in terms of the recoverability. And that is
Mm.
just because the interactions were so strong that I couldn't break them, the interaction between the material and the drug.
Yep.
And we were brainstorming and eventually, like reading papers, discussing with the supervisors. This technique, it's not new.
Mhm.
I didn't invent it. Okay,
Mm.
so yeah, but usually this technique is used for metal ions,
Mm.
things like cadmium
Yeah.
or nickel, that kind of
Mm.
stuff, right? The
Yeah.
use of this technique for or small organic contaminants, I would say something like new, that is still like at the development stage. And
Mhm.
yes, we thought that this could be a good try, and I gave it a try. We had all the tools, hopefully, available in the lab, like like the filtration setup, like a really good expertise with the use of membranes as well. Like
Mm.
my supervisor, Dr. Philip Jessup, he's an expert in the use of membranes. We do a lot of reverse osmosis and nanofiltration, all that kind of stuff.
Yeah.
And I was like, okay, why not? And
Mhm.
then right on the first couple of tries, the thing seemed to work, and I got excited, and I
Wow.
really just dived into
Uh,
the project. Yeah.
yeah. That's amazing that that doesn't happen on often in research where we're
Not
looking to
really, not
survive the
really.
first. So
Maybe a little bit of like luck,
you
I,
luck,
I
yeah.
would say.
Yeah.
Yeah,
Yeah. No, that's great. Yeah, no, and it allowed you
yeah,
to within only a short amount of time create such a great progress on this project. So I'm sure there's going to be
yeah.
much more progress, in a year from now. We'll be hearing all about it.
Oh, hopefully, yeah. Hopefully,
Yeah,
fingers crossed. Yeah, of course.
amazing. Yeah. Um, I'd love to kind of like pivot and just ask you a bit of, well, not really pivot, but essentially something that we've noticed kind of in your background is that kind of collaboration seems to be kind of a big recurring theme, essentially. So whether it be a kind
Of
of
course.
lab mates or young minds, as you know, you're a TA, you've taught, you have a great background essentially. So how do you approach kind of collaboration in your work? And how has it kind of enriched both your research and maybe even a bit about your kind of teaching experience?
Yeah, that was an amazing, amazing question, really, Victoria. Like for me, I couldn't have all these done like by myself only. I really, you know, even like the simplest and maybe silly ideas, those are worth trying. And the
Mhm.
way I work, the way it works for me, I'm a really like, I love learning new things.
Mhm.
I am a really good learner. I'm the kind of person who like bothers people. I ask a lot of questions. I am always seeking for, like a new opportunity to learn something new.
Mhm.
I can even give you one example. I've been to Buffalo in the US
Mhm.
just a couple weeks ago, and it was amazing because I just found a person that works at EPA, the Environmental Protection Agency of the US.
Mhm.
I It was amazing because look, the guy, he was doing essentially the same thing that I do, removing contaminants from water, and
Oh,
I could have the chance to talk to an expert from
yeah,
EPA. It was like mind blowing,
yeah.
the experience. So it's these like small things, like about asking people, showing that you're really interested, right? And in
Mhm.
my case, I really love what I do. I'm always, yeah, seeking for opportunities, and more importantly, I would say it's building like a last long relationship,
Mhm.
and not only looking forward to have like one extra line on your CV, but really, having a nice relationship with people, because, those small, like maybe surface relationships can become something bigger in terms of big achievements. Right now, just the example that I gave you, that I work under this big team here, it's a really nice collaborative environment. I'm constantly chatting with people that do detection. Hey, what's going on, everyone? So do you have any special advice for me?
Mhm.
Engineers, I love talking to engineers to kind of like
Yeah.
have this different way of thinking, because I'm always
Mhm.
like sometimes in my own world, like
Yeah.
thinking about chemistry mechanisms and stuff. But it's nice though when you kind of like has the opportunity to expand, in terms of
Mhm.
like, okay, I'm gonna think outside my little box here.
Yeah.
So yeah, I don't know if
Yeah.
that answers your
It
question.
definitely, most definitely did.
uh,
That's
uh,
amazing that, you had the
I talk a
opportunity.
lot, I'm sorry,
No, no,
Victoria.
no, it's okay. I do too. No, no, you're great. You're really great. Essentially, that's amazing. Collaboration is, one of the most important things, and I'm sure, as you said, you wouldn't be where you would be without collaboration. So, both definitely
Yes.
agree with that, especially in research. So,
Yes, of
um,
course.
yeah. Another question kind of I had, but was kind of a little bit different, but kind of a
Yeah.
little fun one. So I kind of looked into you as well. And you seem like you're very enthusiastic about kind of trying unconventional approaches or even just the craziest ideas. I think you said something to that effect. It's quite refreshing, you're kind of open to trying whatever. Can you give me maybe an example of a time when you consider maybe an unconventional approach and how this might have kind of led to something new or a breakthrough in teaching or research? So something that, random essentially that gave you kind of a new perspective or a new, way of doing what you do. It could be your research, teaching, whatever.
Yes.
Hard question,
That's kind of,
but
yeah, it's a funny, it makes me think a bit more now.
yeah.
Well, I don't know. So, okay, so when I first arrived here in Canada, and
Yeah.
I started my PhD at Queen's, and
Mhm.
then we had like 2 or 3 months to think about what we were going to do.
Yeah.
And I came up with this idea. Okay, so my goal is to remove contaminants from water. Okay, so
Mhm.
maybe the easiest way of doing this is having a fishing rod.
Oh,
It's having a fishing rod. So, and then like the bait would be like some sort of technology, some sort of chemistry that I would just like
unattractive.
I would throw the fishing rod, and
Yeah.
attract the fishes, like the bad
The
fishes,
bad, yeah.
the bad fishes. And then when I presented that idea to the group, everybody laughed. It was funny. I presented it during like the group meetings and stuff.
Yeah,
So, okay, it turns out I didn't develop the like fishing rod per
yeah,
se, just the way I was like imagining.
yeah.
I don't know, but somehow maybe I managed to somehow develop a bait for drugs. So
Yeah.
that is maybe something that comes up to my mind now.
Yeah. No, definitely.
Yeah,
That definitely explains kind of exactly what I was saying, thinking of something kind of
yeah.
out there, a larger picture, and then you realizing that you can relate it to something else. So most definitely, yeah. Um, Essentially, I don't want to keep you for too long. Essentially,
I'm good. If you're good,
yeah,
I'm good. Really,
yeah.
yeah.
Yeah, no, it's up
Yeah.
to you. So I might have kind of one or two more questions, but if it's up
Yes.
to you if you'd
No,
like
that's
to.
okay, please.
Okay, I mean, another
Yeah.
kind of question, related to that would be kind of about the field that you're in. So, what do you think are some pressing challenges in the field of kind of environmental chemistry? And obviously, kind of how do you see your research contributing to addressing these concerns? So, where do you see kind of what are the biggest challenges that you kind of see in the whole field, essentially?
Yes. Okay. Yeah. I mean, technically speaking, I believe, well, I do research on the removal of contaminants of emerging
Contaminants,
concern.
yeah.
Okay, so we have this big challenge now that is like the ubiquitous presence of PFAS,
Yep,
right? So
yep.
PFAS, so they are a
PFAS,
really big thing. Yeah, they are a
yep,
big theme now.
yep.
So I would say that, because this what happens? Okay, uh, in the first place, why do scientists develop chemicals that cause harm to the environment,
Mm-hmm.
right?
Yep.
So that is
What
like,
firefighting foams and certain things have PFAS
yeah.
in them.
So
Yeah.
why don't we develop, if we need like a chemistry or something that is gonna, bring benefits to the
Mm,
humanity? Why
yeah.
don't we develop something that, since from its beginning, it is
Mhm.
something good
Yeah.
that's not going to cause harm to the
Yeah.
environment,
Yeah.
right? So, sometimes I read a paper and the guy, oh, this is nice, it works perfectly, but the way the thing was synthesized or prepared, it used a lot of crazy expensive chemicals
Mhm.
using super high crazy temperatures, and the recovery uses sulfuric acid, and I was
Mhm.
like, man, no, this is not right.
Yeah.
So I believe, for me, especially that I do green chemistry, is it's
Mhm.
really thinking about your own way of doing your research in terms of like, okay, um, is my strategy really sustainable?
Mhm.
Because I'm claiming that I'm helping the environment,
Yeah.
okay? So every single step during this process has to be, uh, has to kind of like fulfill with these requirements
Mhm.
throughout the process. It has to be sustainable since day one, because if one
Yeah.
of your stages causes a lot of damage to the environment, so I don't think your process as a whole is sustainable,
Mhm.
right? So this kind of knowledge, I don't know if maybe not, it's not all researchers that have this way of thinking,
Yeah.
about evaluating themselves in terms of like, okay, am I being sustainable on
Mhm.
the first step, on the second step, or am I just claiming that it's my final product that is sustainable?
Mhm.
Okay, so that goes around, Victoria. That goes around practicing more sustainability.
Yep.
So that for me is a really big challenge in terms of developing a technique. And on the side of what we have right now, the PFAS, it's a big
Yep.
issue that,
Yep.
yeah, we need definitely collaborations and,
Yep.
you know, teaming up with experts in the field. Yeah,
Yep.
so I would say about those two things, maybe. Yeah,
Yeah. No, that's great. That's great. Yeah, we've been
yeah.
I've been learning about PFAS a lot recently. It's been really coming up in
Mm.
the recent trends, and more countries are kind of developing their own regulatory kind of, you know,
Yes.
how much each country is deciding is okay in water. And it's really eye-opening, to learn that this has been going on and they haven't, it's been, only brought in recent events almost. So it's great that, you know, the work in your field is doing something to kind of countervent this, and that you see there being an issue here. So great to hear. But yeah, essentially I don't have many more questions for you.
Maybe
You can,
if that's okay, maybe something that I forgot to mention is just
yeah, of
because
course.
I usually don't talk about those things when I'm presenting the posters
Yeah.
and stuff. But
We're open to hearing everything, not just about your research, about anything, honestly.
awesome. Yeah,
So
that's great.
yeah,
Thank you so much for making yourself available. Yeah.
yeah.
So, okay. Two important things. One I've already mentioned is we have this big network at Queens, right? It's
Yep.
the CCREN, okay? And also my research, it does align with some United Nations sustainability goals. So the UN sustainability goals, right? So for example, why am I telling you this? In my research here, I am not trying to solve the world's problem to remove all drugs in the world. No, it's not. That is not my goal. My goal primarily is to make clean water available, especially for people that, for example, don't have access to a water treatment system or something like that, right? So overall, you know, during my recycling here, I really don't care too much about the thing going back to the environment because we have other people that do the destruction of those chemicals, right? So as you can see, all these processes here, they are physical processes. So I don't do like chemical, I don't do breakings of chemical bonds. I really don't appreciate that idea because, of course, we have a lot of amazing chemical techniques, but usually they break chemical bonds, and the smaller pieces that are generated when you break the original contaminant, right? We don't know whether those smaller pieces are even more harmful than the original contaminant.
Yeah.
Right? So, okay, so in my research here, basically, okay, I have this dirty water, I don't know, contaminants, whatever, and I apply my technology, and then I get clean water out of that. Okay, so that's and then this aligns with the UN goal number 6, more specifically. So I always have these kind of like, I try to keep within the range.
Yeah.
You know what I mean? Yeah. So maybe something else to share with you.
Yeah, that's definitely interesting and good that you're, you know, looking to how United Nations is doing. It's good that you have a goal essentially that you're working towards as well. So that's great.
That's right.
Yeah.
Yeah.
So unfortunately, our Zoom call is we only have like a couple minutes left. So I did just want to say, you know, kind of like an outro. So, you know, obviously, thank you so much for speaking with me today. It was a really great conversation, and your research is going to be very impactful, I see. So I just want to say thank you so much, and we'll definitely keep in touch. I'd love to hear more about kind of where it goes in the future and how, you know, it's progressing. So love to hear
Yes, for me, really, it was such an honor. Like I'm a super bad liar, right? So really, well, I was kind of amazed that I got the email from Jade first, and yeah, super happy. Anytime I'll be around. So if you're here in Ontario, whatever,
yeah, we're actually
I'll be
in
at Queen. Oh, nice,
so close,
awesome. So I'm here in Kingston, so Queen's University doors are opened.
Yeah.
Right, if you want to come and visit us. So yeah.
Well, very interesting. Yeah, thank you so much. And I will definitely be in touch regarding kind of, you know, next steps, and I'll send you everything, see if you're okay with it.